Betty Olson, and I'm going to talk to you about something that people have been discussing ever since I first graduated with my doctoral degree. So fecal coliform is a term that's near and dear to my heart, and I'm sure that over the next few years you'll get to be extraordinarily familiar with it. So with that, I'm going to begin. So we have an outline. The first thing I'm going to talk about in this presentation are what an indicator means for public health. And public health, of course, has to do with body contact and with swimming and protecting people who have drinking water sources. I want to begin with total coliform because, in a sense, fecal coliforms seem to be directed down the same path that total coliform have followed. So I'll begin with their definition, some of the issues that arose in drinking water, and then I'll move on to the fecal coliform and talk about methodology and sort of where we are now. So an indicator, as you can see above, we have a general definition, which is a biochemical marker that occurs at the same time as the constituent being examined. So this is a very broad definition. But when we change to an indicator of public health, we can see that we want it to be a member of the normal intestinal flora. We want it to be exclusively of fecal origin. We would also like it to be found only in humans. So if we're interested in human disease, we know that most of it is associated with organisms that come from humans themselves, but we have been seeing a change over about the last 3 decades. So we would like it to be absent when the pathogen is absent, in higher concentration than the pathogen, resistant to natural environmental conditions in water and wastewater treatment, slightly better than that of pathogens. So we want it to tell us when if it's that gone, the pathogen should be gone. We'd also like it to be easy to isolate or to test, to identify and enumerate. And we'd like it to be non-pathogenic so that we don't have to worry about the indicator itself as being pathogenic. So what you can see in this slide is we're looking at the different groups. So coliforms is a very large group of organisms. Within it, we have a subgroup called the fecal coliforms. Within that, we have a group of organisms called E. coli. And within that, we have a group of pathogenic E. coli called the diuretic E. coli. Now, since about 1980, this group keeps growing. Many of them are associated with animals. Many of the ones associated with animals are strictly infecting and causing disease in animals, but they also cause diseases outside of the GI tract. In rabbits, for example, it's a lung disease, and in domestic dogs and cats, it can cause urinary tract infections, just to give you one example. So if we look at the definition of a total coliform, we can see that they're commonly found in the environment. So we see them in soil, vegetation, and they're generally harmless. So the vast majority of these, unless you're immunocompromised in a severe state, will not cause a health problem. Total coliform bacteria then are detected in drinking water. It means the source is most likely environmental. Fecal contamination is not likely. However, it means that you do have environmental contamination entering your drinking water system, so it's important to find the source and to resolve the problem. Now, I took this off of the state of Washington's site so that I'd be sure to be thinking along the same manner as your site. But I wanted to talk about some of the problems that came up with total coliforms. Originally, they were the sacrosanct measurement in drinking water systems. But in the 1980s, we found out that there were many drinking water systems that began to have continuous coliform outbreaks. And this caused a large problem since the 1974 Safe Drinking Water Act had required utilities to notify the public and required boil water. So many utilities that could not find any source for these coliforms that was of public health significance were forced to have boil water issues. They also then certain ones added up to 12 parts per million, ended up with residuals of 12 parts per million total combined chlorine. This had a negative effect on their customers. At that level, it bleaches clothes, evidently, but also it creates a lot of disinfection byproducts. So the EPA at that point began to rethink this, and there was a lot of research done, and they found out that coliforms could survive in drinking water systems within a biofilm, even when you had a high disinfectant level. And then, as they would grow out of that biofilm, they'd break off and you'd get a positive test. So it became quite clear it was not a good indicator of public health, but it was a good indicator for a high organic load because they cannot survive unless they have a significant amount of nutrients present in the water. So that brings us to the fecal coliform. And the fecal coliform are a subgroup of the total coliform bacteria, as you saw in the diagram. And they exist in great quantities in the intestines and feces of both people and animals. The presence of fecal coliform in drinking water sample often indicates fecal contamination. But you see often, and often is an important word in this case because there are instances when you have fecal coliform indication when they're not truly from a fecal source. So the meaning then is that there's greater risk that a pathogen is present if you have fecal coliforms, and only fecal coliform bacteria is detected. So those are kind of the byways that we go. So how do we get fecal coliform as an indicator? So we got it because you can see here, watch out for the cow patties. This is, and then birds, of course. And we see our good friend dogs, especially in urban areas, when their owners don't carry their plastic bag. So we see the term fecal coliforms has arisen from trying to find a rapid, dependable method to detect organisms that establish the presence of E. coli or closely related variants. And they don't want to go into any type of pure culture. You just do the test, you get a read, and you know whether or not you have fecal coliform present. So the methodology then is important because fecal coliform, it's not that you identify a particular bacterium that's known to come from feces, but it's actually a definition of a test. And that means any bacterium that is gram-negative, not sporulated, which means it isn't in a resting stage, has a certain type, oxidase negative, meaning it doesn't oxidize hydrogen peroxide, and can live aerobically or anaerobically, and is able to multiply in the presence of bile salts, which inhibit other groups of organisms that could interfere with the test, or other surface agents, and have equivalent properties and are able to ferment lactose with acid and gas production in 48 hours at a temperature of 44.5 degrees. So in a sense, this is a temperature-dependent test. Any organism that's able to grow at 44.5 and satisfies these categories will show up in a fecal coliform test. So for example, if you have bacillus and it hasn't formed the spore stage, it sometimes can withstand the bile salts and give you a false positive. So you don't always get fecal coliforms, and that's why you have confirmatory tests. The other factor is it's not a numeric test in the sense that when you measure a chemical, you are measuring a quantity such as micrograms per liter. It's actually based, oftentimes when tubes are used, it's based on the most probable number. And the reason why I bring this up is because it is an estimation. It's a probability measurement. And since it's a probabilistic measurement, it isn't bounded in the same manner. So we can see here, and what I want to show you is with the tube method, how labor intensive it is. So you can see they've done a number of different dilutions here, and the test wants to have 3 to 5 replicates and 3 different dilutions. And then you get a number and which you want bounded on each side. So you want one that's all positive above it and one that's all negative below it. And then you read that, and that is an MPN number. Now, what you do with that number then is that you go now to an MPN calculator, which is very nice. In the old days when I started, you had to go to a book and read the tables, and then you find out the tables sometimes had errors in them. But most importantly, they have very, very wide confidence intervals. So if you find, for example, 15, it may be from less than 2 up to 1,500 would be the confidence interval. So it's much different than dealing with a chemical in this probabilistic manner. So there are problems with the test too, and you see that temperature is a big problem. So if you take an organism from the environment and plop it in 44.5, for example, from Lake Washington, it might not survive because they have bacterial injury from being in the environment. So there's a temperature, and then there's also selected media, both of which cause problems in your ability to be able to recover all the organisms. So we can see then we have a method for coliforms where we try to get more precise. This is a membrane filtration method. Now, of course, they come in easier setups than these, but you have your filter, which is supposed to trap all bacteria. So a 0.45 micrometer membrane will trap the fecal coliform on it. We have a specific medium called the MSS medium, and we also have a medium called the ME, or the Enterococci. So in marine waters, the EPA recommends looking at Enterococci. In fresh water, it recommends either Fecal Coliform or E. coli. So you can actually count these. Now you have a concentration, and it has better confidence intervals. So the advantage is obviously you have an exact count or an exact concentration. So this piece I wanted to show you what they look at. So here we're looking at, these are coliforms, and the correct coliforms have a metallic green sheen, which you can see here, and they have a modified endo. So you still can measure these organisms. California has, we have guidelines, and we have coliforms, Enterococci, fecal coliforms, and E. coli, so it's an indicator of your choice. So over here we see the fecal coliform and MFC, and you can see that they're coming out blue, and that the incubation is 44.5. Sometimes you get this, which you can see is difficult to count, and you want to confirm at least 10% of the organisms in either case. There's a new method, which is a presence-absent method. There are lots of different types of media that you can buy commercially for this, but it gives you the coliform test, and you can see a positive one here, and that's the fermentation of lactose within 24 hours with the production of acid and gas. And the enzyme the coliforms have, and fecal coliforms as well as E. coli, they all have the enzyme beta-galactosidase, which allows them to use lactose sugar. And then another enzyme which is specific to E. coli, glucuronidase, and they break down the sugar glucuronidine. And the idea of this was it's very specific to E. coli. But like everything in the world, it's not perfect. So if you look, you can see that Bacillus has it. And also, if you think about E. coli, and we're interested in E. coli 0157H7 from a public health standpoint, that organism does not have this enzyme, so it will be negative in this test. So nothing is perfect, but the vast majority of E. coli are positive, and you can see that they put a fluorescence, so you just get a fluorescent sign, so it's easy to tell a positive. So we have some issues with fecal coliforms. So we know that they have several different genera that are present, and they're not all of fecal origin. So we have them in plant materials, pulp and paper, mill effluents, that also meet this definition, meaning they're thermotolerant. So we see they can belong to Klebsiella, Enterobacter, Citrobacter, and some other, but these are the main ones that you find. And Oregon in the 1980s found Klebsiella was causing many positive fecal coliforms in drinking water because it was growing on redwood. Now we know that they're associated with the phloem of redwood, and they also will go up into the vascular material, for example, of a number of plants, like lettuce plants. So these are false positive indicators of fecal contamination. And these organisms can grow in the environment under natural conditions. And I think some of our other speakers will be giving some examples of both fecal coliform occurrences and also perhaps some of these other false positive. So they occupy non-fecal niches in water, food, and waste. So we've learned then that they're not as specific as we used to think they are. And so in 1995, when the news media reported that the highest populations of fecal coliforms ever found were found in restaurant brewed tea, there obviously was a brouhaha. But it turned out that this group was exactly those that we had been talking about, these Klebsiella and Enterococci, but no E. coli. So they got fecal coliform positive, but not E. coli positive. And so when we look at runoff, we can say, where do all these fecal sources come from? So they obviously, a large part comes from agriculture, but in all the source tracking studies I've been involved in, oftentimes it is indeed birds. So you see birds, and then obviously homes. Septic tanks can be a huge problem, as are also sanitary sewers if they're not taken care of. So in a watershed, you have not only these types of animals, but you have a broad range of wild animals that can contribute significantly to the fecal load in an area. And the question is, for you all today, how do you really control that load, and what is its significance in terms of public health? So I wanted to take an example closer to home, and I found this article from September 9th, which is for Sanish Bay in Oregon. And I must say that my son just graduated in June from the University of Oregon. He had a great time, and he grew to love the Northwest. So he went to many different beaches, but you can see how beautiful your countryside is. But they also had a problem with fecal coliforms. And so they say the culprit is fecal coliforms, and you can see livestock, birds, and of course humans. So you can see septic tank issues. And then lastly, as we move forward into this talk today, we need to ask what the major goal is. So there's a goal of public health. And that's a very important goal for all of us to adhere to. And certainly for myself, with my background, I've always feel how important it is. But we can also be concerned with nutrient input. And so fecal coliforms influence that. We can also, therefore, be concerned with reducing the sources of runoff, period. So other than water, what comes in, that could be our goal, to reduce that. And differentiating dry period runoff from wet period runoff. We found in Southern California that they were very different, and also in Northern California. And you could find out different types of information by examining dry flow runoff versus wet flow runoff, in terms both of fecal coliforms and the quantity, but also in terms of the type of fecal sources of input. But I want to end this page by saying that the EPA, in its latest guidelines for beaches, says that there's no association with fecal coliform and swimming and GI disease. So that's an important point to make also. They find that Enterococci is a better association with disease in seawater and E. coli in fresh water. So now I want to give you an example, and I take this example from the state of Georgia, and it's a few years older, because Georgia started looking at the TMDL for fecal coliforms in the late 1980s. And you can see this is a case study from 2003. so that it is obviously about 8 years old, but it'll give you an example of some of the things that you might run into. Georgia is also quite a rural state, so it does have a lot of forested areas, and it has a lot of animals. When I went back to work with them, also CDC was involved, and Georgia was very concerned about its ability to meet a fecal coliform level of 200 per 100 ml, and I realize under certain circumstances, the one in the state of Washington is more stringent. So when we look at this slide, we can just see that the light brown areas are fecal coliforms and others, and that was 62% of all of their streams gave positives for fecal coliforms. The ones that they found that were fecal origin only were 44% of their streams. And so this led them to say, well, for us, maybe fecal coliform isn't the best measure for public health, because they were primarily interested in public health. And so they decided that they would like to use E. coli. And you can see in 2003, they still had a TMDL based on fecal coliforms, and it took them a number of years to change the way it was done from the state point of view, legislatively. But they felt this was their best way to in terms of their resources, but as I say, their goal was public health. So I wanted to just end today by talking about E. coli. So E. coli is a subgroup of fecal coliforms. Most are harmless, and they're in the intestines of warm-blooded animals and people. And some strains, as I mentioned earlier, do cause illness. So the presence of E. coli in the drinking water sample almost always indicates a recent fecal contamination. And I can tell you, in looking at probably more than 1,000 drinking water samples, I've only found one positive for E. coli. I found many that were positive from coliforms. We found out that people had worked on the pipes. So although they followed the AWWA standard to disinfect at 50 parts per million, they still had coliforms that survived. So the meaning is that when you find E. coli, there is a greater risk of pathogens being present. But all fecal material does show some risk of pathogens being present. And the presence in runoff represents certainly fecal material, and you can't have these thermotolerant interfering coliforms. Lastly, I'd just like to say one thing about microbial source tracking. There are many, many different ways of doing microbial source tracking. This is something that we did for Monterey County, for Watsonville Slough area that had, it was a similar type of situation where the slough was being contaminated, and they looked at wet and dry periods. So I'm only going to talk about cows and birds, but if you see here, the high parts are in the wet periods for cows, but in the dry periods we saw no cow indicator. So that meant it was being washed from a fairly significant distance away from the area, and in our part of the world, there isn't rain oftentimes April through November, this year being an exception, and birds. So as I mentioned before, birds are a major contributor to all fecal sources in water areas. So just to leave you with a little bit of, this tells you, and it's in your printout so you can have it for reference. I'm not going to go over it here. But you can see the numbers and the different types of bacteria that are in fecal material. You can obviously see why fecal coliforms were chosen. They go up to 10 to the 9th per gram of wet weight of fecal material. And then I thought I'd just end with showing you fecal coliform and fecal streptococci in a variety of different animals. So with that, I'd like to thank you for your time, and be glad to entertain any questions if you have them.